mean, how confident you are with the model that they have generated. They will give you some confidence score. Okay. All right. So yeah. So remember that you are going to provide the sequence, right? And you are going to get the template from the database, structured database, right? So the accuracy of the homology model, they are strongly dependent on the sequence identity between the target, right? Target and the template. Okay. So there is something called sequence identity and sequence similarity. So in the case of homology based modeling, we are considering mostly this sequence identity. Okay. So in what do you mean by sequence identity? So you have, let's say, um, uh, tryptophan, right? So tryptophan is there, and another, if this is in the target structure, okay, target sequence, and the template sequence you have here. So when you align, you are finding that tryptophan is aligned with tryptophan, right? So this is exactly the same. So this is identical. Okay. And then you have another tryptophan, but you have tyrosine, right? So they are not identical, but since their properties are the same, they are both hydrophilic, right? Hydrophobic, actually. So they aromatic and hydrophobic, right? So they are sequence similarity. Okay. So this is sequence identity. So we are looking for this one, not this one. Okay. So, but when you align, you will also see the sequence similarity, but we are considering this. Okay. So when you have sequence identity of greater than 50%, okay, that means the identity of the target and the template, right? So if it is more than 50% sequence identity, you can produce the model very accurately, and reliable model you can generate. That means you have good backbone and sidechain prediction, right? So if you have like about 30 to 50% sequence identity between the target and template, yeah, so the model you generate will be in average, and you have to, be very careful. Okay. So requires validation, and if there are loops there are missing, or when you align, there may be section of the regions where there may not be able to generate the model. So there may be missing loops and all, so you can again generate those. Okay. And if you have the sequence identity less than 30%, okay, between the target and the template, they are now considered in the twilight zone. Okay. So you have to be very conservative. Okay. So you have to be very careful in this kind of structure, right? So everyone with me till here? Yeah. Okay. So should we consider this sequence identity only to, select our template? Of course not. Okay. So it depends. Again, I'm going to tell you that whatever, you know, technique you are applying in your study, like if you are applying docking also, or if you are applying homology modeling, if you are applying MD simulation, anything, okay, you need to first understand what you are trying to achieve. Okay. So here in homology modeling or 3D structure prediction also, you have to decide on based on your objective. Okay. So if your objective is to study like the whole structure, dynamics of the protein structure, okay, then the template selection will depend on your objective. Okay. And if you want to just concentrate on the active site pocket for doing docking, again, your template selection for modeling will depend on that particular objective. Okay, so I will explain what I'm trying to say. Okay, so like you have this target sequence. So you have this target sequence, amino acid number 1 till, let's say, 200. Okay, when you search this sequence in the database containing known structure, okay, like PDB database, okay, what you are going to do is sequenced alignment, okay, because you are trying to find homologous or evolutionary related sequence against the database for this particular target sequence, so that because this sequences in this database has corresponding experimental structure for all of these sequences, right? So using that particular uh, related or homologous structure, you can build the model. Okay, but you see how to select a particular template. Okay, so let's say when you search in the database, the first or the top scoring, whatever, yeah, based on identity or some score they have given. Okay, so in class, if you are doing, you will be giving blast score, right? So whatever the score they are giving, let's say you have this particular first hit. Okay, so let's say from the template, this is the first hit from the database. Okay, from the database, and this is the second hit. Okay, and this is the, let's say, third hit. Now let's consider that this is the alignment. Okay, and the first, I mean the first hit, when you aligned it, it has around 60% sequence identity. Okay, so based on the previous slide and based on that particular criteria, obviously you will be selecting this one, right? The first one. But there are some consideration you have to take. Okay, so I will tell you that. Okay, so again, you see the second template. So in the case of the second template, it is not matching the end terminal. Okay, this terminal, this region, but is matching the C terminal, right? And let's say in the C terminal where it's matching, it is having like 40% sequence identity. All right, and this one, from somewhere it's matching here, somewhere it's matching here, let's say it's having 30% sequence identity. Okay, fine. So based on the sequence identity, you will be inclined to take the first hit, okay, from the database. But okay, just consider this. So you are going to, you know, your objective is, build the structure, model structure, and do molecular docking. Fine. So you are now concerned where is your functional domain. Okay, that means the catalytic domain, which is very important for your protein function. Okay, so if the functional domain is also here, then you are good to go. Okay, you can take this particular template and start building the model. But if the catalytic domain for functional domain is present here, okay, and if you take the first template based on the sequence identity, you will be able to model the protein from here to here only. So your target sequence is matching from here to here, but the functional domain is present in the C terminal, let's say. Okay, so this is not there. This functional domain is not here, and it is present in this region. So you will be missing the active site pocket, and you will be just building the or constructing the model which doesn't contain the active site pocket. So what kind of docking study can you do from this, even though you have sequence identity of 60%? Okay, so model building, it's all depends on your objective of your study. Okay, so now in this case, I will consider this template 2 because the functional domain or the catalytic pocket is present in this N terminal, and the second hit is covering the this particular region. That means I can model the particular pocket from this template and I can do docking. Okay, so now there is another concept called sequence coverage. Okay, so even though you have 60% sequence identity, the coverage is, let's say, 60% only, like 70% here. Okay, and you should see, okay, if the sequence coverage, okay, the same thing only, but they have given just a term. Okay, so like sequence coverage should be also under your consideration, same meaning only, but when you have to do modeling, you have to consider like how much is the sequence coverage also. So in this case, you can take this, like even if you have this 50% sequence coverage or 40% sequence identity, since the functional domain is present in this region and this particular template is able to cover or contain this functional domain part, I will be uh, you know easily choosing this second template to do the model. But in some cases you want to consider, you are not, you don't need this active site domain. Okay, you are not, your objective is not to study docking or do the functional kind of analysis or kind of docking. You don't want to do that. Okay, you just want to see conformational changes. Let's say this particular protein, when it interact with some other protein, how they are changing. Okay, the functional conformationally how they are changing. You can do molecular dynamic simulation to see the changes for that. So you want a complete structure to model, right? So you have another template for the hits here. Let's say that you have, even though it has a huge coverage, okay, but the sequence identity is somewhere around 35%. Okay, so based on your objective of the study, right, so I would be inclined to take this particular hit because it is covering the whole structure of the protein, and I want to study the conformational changes of the particular protein or the particular target protein. So I will be considering this particular uh, template. Okay, so there we are not going to do multiple template uh, alignment or building the structure with multiple template, but you can also build that model using multiple templates. So you can take whichever template which is able to cover the whole protein. You can take 1, 2, 3 templates combined together and build the model from this combined template. Okay, you can do like that. So important takeaway from here is you don't consider only the sequence identity, you don't consider only the coverage. Okay, so you have to consider all these criteria based on your objective of the study. Okay, so yeah, so if you understand this. No, template. Okay, so seems like you have missed the first template, first slide, right? So template is the s- structure or homologous sequences present in the database. Which database? The sequenced i- in this database, it also contains sequence, but the sequence have corresponding experimental structure. Okay, so you are going to search your target sequence, which you are planning to construct a model against this database containing sequences where corresponding structure are present. So if you are able to find similar sequences from this database, okay, then the hits that you get, okay, the high scoring hits that you get based on sequence identities, coverage, or any other scoring uh, score, okay. So you have to decide on which hits you have to take, okay. So the particular hit that you are going to take for model building is called a template, okay. It's not the ligand, okay. I'm not talking about docking and getting the hits. Hits means anything that you, when you search, when you sort it out, the top scoring, whatever results you get, you can call it as a hit, okay. It can be template, it can be ligand, but in this case we are talking about this structure that is present in the database, okay, which are having high uh, identical score or the high coverage depending on your particular study, okay. So I hope that with this we can start doing experimental or hands-on session, okay. But before that, we are going to consider this example, okay. So there is something called Bepsipen. This is a cysteine protease. Oh, this is wrong, okay. This is from Piki. Okay, this is an organism protozoa, right? So this is what is the biological role of this Bepsipen. So this is a cysteine protease and belongs to the papain family of protein, right? And expressed by this particular organism. It's a protozoan and it causes the bovine babiosis, right? So it is involved in the degradation of the host hemoglobin, okay. So it breaks down the red blood cells and it provides amino acid critical for the growth of the uh, and replication of the parasite, okay. So what is the relevance? So see, always you have to go back to your target protein or the target enzyme that you want to study, what you are planning to do, okay. So you see the relevance of this study, okay. So Bepsipen shares structural similarity with falciparum-2, Plasmodium falciparum. So it's a validated drug target, okay. So when you design drug for Plasmodium falciparum, you go for this falciparum, okay. So falciparum belongs to the same family of protein, this papain family, okay, the cysteine protease. The same with the falciparum, we have this Bepsipen for this particular organism called Bepsia begemia, right? So what we are trying to do, inhibition of this Bepsipen function. So we are going to inhibit the function of this particular protease, okay, cysteine protease enzyme, that is Bepsipen, which has the potential to block the parasite access to nutrients, because it won't be, if you block this particular or inhibit the function of this particular enzyme, it won't be able to degrade this or break down these blood cells or the hemoglobins. Uh, that essentially it means that the parasite won't be able to replicate, right? Because the nutrients won't be there, right? So disrupting its life cycle, right? So this is the whole story. So we want to inhibit the function of this particular Bepsipen enzyme, okay? So what are the catalytic sites? So you may not find this in your target structure or your target protein, but if it is an established or validated drug target, you will be easily able to find this active site residues, okay. You can do a multiple alignment, okay, and see where the conserved residues are based on the study of previously validated protein. Okay, in this case, we did a study on falciparum 2. Okay, and some other established organisms. Okay, I mean established enzyme, this papain family of enzymes. Okay, and then we come up with this particular catalytic region. Okay, this forms a pocket. Okay, so cysteine, this is a cysteine protease, and cysteine particular residue initiate the nucleophilic attack, and then it this histidine forms a base, and this asparagine stabilize the particular reaction. Okay, so this it forms a catalytic triad. Okay, so these residues form a deep hydrophobic clay optimized for peptide bone hydrolysis in the globin chain. Okay, so like, yeah, anyway, so let's not go into this too much. Okay, so I'm just telling you that when you build a model, you should know where your particular functional domain is. That in this case, you have to find out where are the catalytic residues are. Okay, so that you can decide on the template that you want to select when you search for the when you search the sequence in the structural database. Okay, so you have this important thing when you are after the constructing the structure. Okay, so you can do all those things. So what are the drug design implication in this case? So define the substrate binding group and conserve catalytic geometry based on geometry, make Bepsipen suitable for the structure based inhibitor development. Okay, so once you have this structure model, you can go for finding the inhibitor of this particular enzyme. Okay, so in a simple way, you can do docking and see if you can find inhibitor for this. Okay, so selective targeting of the Bepsipen could lead to novel parasitic agent for babesiosis, especially in drug resistant cases. Anyway, right, so the our objective is clear, right? So we need to model the catalytic site. We are interested in the catalytic site because we want to find the inhibitor for, or we want to do docking and find a new inhibitor for this particular cysteine protease. Okay, so that we can find some kind of treat or babesiosis. Okay, so let's start with the hands-on session. So I've given you the link for downloading a modeler. Okay, so we'll be using modeler first to build the model. So everyone, you have downloaded the modeler and install also. Yeah, okay. So before downloading, I mean showing you with modeler, we have to first